Hi, my name is Dr. James Woods. I'm here from Jordi Labs, and today we're going to talk about 510(k) submissions. So, first of all, we're going to go over who is Jordi Labs. Secondly, we're going to cover what's necessary for a medical device submission. Thirdly, we're going to cover where does Jordi Labs fit in with your medical device 510(k) submission. And finally, we're going to cover not only some example analyses, but also how we can accelerate your testing and your submission. Founded in 1980, Jordi Labs is initially a GPC analysis lab. GPC is a way to determine the molecular weight of a polymer. Since then, we've really blossomed and become a full-service polymer analysis lab. As part of that, we conduct extractables and leachables analysis, analysis in support of regulatory filings, medical device screening, and investigative analysis. We perform over 1,000 projects annually. More than 80% of our employees at Jordi Labs are degreed chemists, and we really pride ourselves on high-quality results. So I guess to the topic at hand, suppose you want to sell a medical device. How do you get to market? In the United States, there are several ways that you can do this, and the first thing that you have to do is you have to review public data for similarly marketed devices. You have to take that public data and review how that device is going to be regulatorily accepted and determine the regulatory pathway for that. Secondly, you have to review the guidance for recognized consensus standards. For example, if you make a device that the FDA has already regulated, they have guidance documentation on their website that's available to you that would tell you what sort of testing they're looking for when you go to perform testing. And then finally, you can review that guidance document and determine your device class. There are many different kinds of devices, but the FDA has pigeonholed them into different categories, and different categories have different requirements. And we'll go over that in a second. For example, Class 1 devices are considered low risk. Things like Band-Aids, things like crutches, those are considered low-risk Class 1 medical devices. Moderate risk is associated with Class 2 devices, and the FDA requires things like general controls, which are the controls that go into manufacturing and other process controls that go into that to regulate those devices, but they also require special controls, for example, the 510(k) process. For devices that have a significant risk, where they're either life-sustaining, like a pacemaker or another sort of device that helps sustain someone's life, and there is a significant risk that if the device fails, it could cause patient harm. The FDA requires not only general controls, but also a pre-market authorization, where you as a medical device manufacturer must prove safety and efficacy for the device. And so, as a few examples, Class 1 devices, elastic bandages, crutches, wheelchairs, bedpans, things that it doesn't necessarily make a whole lot of impact on the patient outcome if the device accidentally fails. On the other hand, Class 2 devices, things like infusion pumps, contact lenses, they're in contact with the patient. Tracheal tubes are another example, hip prosthesis, for example. Class 3 would be things like tissue adhesives, things that are implanted in contact with blood or brain tissue. Mechanical heart valves are another example. And so there's a couple of examples on the slide to take a look at. So based on the classification of your device, there are 4 routes to selling a medical device. We just discussed the pre-market approval, the PMA process. The PMA is a route for when there's no existing equivalent predicate device. So this is brand new territory. We've never done it before. It requires prospective and randomized clinical trials, and these are trials in humans. In contrast to pre-market notification, if there's something out there that has kind of broken the ground for you or is one of those existing approved medical devices, you can show that you are substantially equivalent to that in terms of safety and efficacy. It does not generally require human clinical trials. And there are several other exemptions, for example, a humanitarian device exemption for small patient populations, and then also investigational device exemptions if you're doing something as an intermediate approval en route to a PMA. There's a couple of restrictions, for example, on the HDE, the human device exemption, where the final cost of the device has to be under $250, and oftentimes it's either the lower it's the higher of the two, either the cost of the device, the actual cost, or $250, and it's not effective. So basically that takes those 4 routes to getting an approved medical device down to 2 routes for an approved medical device. Either the PMA, which requires human testing, or the 510K process, which doesn't necessarily require human testing, but it does require that there's something substantially similar out there. And so the numbers that I'm showing you right now, in 2015, the FDA approved 47 original devices under the PMA process. In contrast, they approved over 3,000 devices through the 510K process. And so what I'm trying to get at here is a 510K is pretty much the route to go for your medical device. And to illustrate that, here are the differences between the 510K and the PMA programs. So the 510K demonstrates substantial equivalence to a predicate device. Once it's gone through the process, the new device is FDA cleared. And this is in contrast to the PMA process, which once it's gone through the PMA process, that device is then FDA approved. And so it's a very big distinction to make for these two processes. The FDA response for a 510K is typically within 90 days. In contrast, the PMA process can be done within 180 days by statute, but on the FDA's website, they even tell you that they're not going to get it done in 180 days. The documentation for a 510K really only compares the novel device, your new device, with the predicate device in terms of safety and efficiency, or sorry, safety and efficacy. And so for the PMA, in contrast, you have a formal clinical study that involves patient populations. It also requires laboratory studies. The 510K doesn't necessarily require human data. And so there's a big advantage in terms of going through the 510K process as opposed to the PMA. In summary, 510Ks are cheaper, easier, and faster. And so now that we've decided that we're going to do a 510K, the goal is to demonstrate substantial equivalence. And the key elements that the FDA uses straight from their guidance documentation, the substantial equivalence of the predicate device, the intended use, the technological characteristics, and the performance. And so these are the metrics that the FDA is going to use to assess whether or not your novel medical device is substantially equivalent. They have this excellent flow chart. I believe it's in Appendix A of their guidance documentation that helps illustrate where Jordi Labs can fit into your 510K. And so I've highlighted in red those appropriate questions. The technical characteristics are a key component of where we can assist you. The material characterization, for example, or the device extractables and leachables profile. Additionally, we can help with performance characterization because they're not only interested in what gets left in the patient as the device is implanted in someone, they're also concerned with how it's going to perform over time. And I have a case study at least where we cover a little bit of that. So we'll get to that in a moment. Here we are. So here's the example case study. For this, it's a hip prosthesis, and that's a Category 2 device. And so in this particular case, there's an acetabular cup that is implanted in the patient's hip that serves as the acceptor for a ball. And there's a plastic liner between the ball and the acetabular cup. And then there's a femoral head and a femoral stem, which is then implanted into the femur. And there's a nice picture of our acetabular cup. For this particular example, we had a predicate device, which was a hip prosthesis that used regular high-density polyethylene. And for this particular hip prosthesis, they used highly cross-linked, ultra-high molecular weight polyethylene. And so the first step for this is to review similar devices to identify the device class, the product code. The FDA maintains a searchable database of every accepted and submitted 510K. And so from this, you're able to identify relevant regulations, the classification panel for this, and possible predicate devices. And so in searching the FDA's publicly accessible website, here are the most recent similar devices. They all start with a K number. For example, K162303 is just one example. And what the FDA documentation on their website offers is not only device-specific guidance, but also cross-cutting guidance for many different device classes. And as always, the FDA favors recent predicates. So if you're getting something from the 1970s and trying to compare that to something that was generated more recently, they're going to ask you to go back and look for more recent predicates. And so here are just a few examples of some similar devices. And so taking a look at just one of those predicate devices, the commonalities in this are that it's an aluminum zirconium-based ceramic femoral head, so it's a nice, shiny, smooth surface with a titanium substructure. It has an acetabular cup and a femoral stem. The differences, which are the significant component that the FDA is going to look at, is that the new device has a highly cross-linked ultra-high molecular weight polyethylene acetabular liner. In contrast, the predicate device used a linear ultra-high molecular weight polyethylene acetabular liner. And so the difference between the highly cross-linked ultra-high molecular weight versus the linear ultra-high molecular weight, I mean, you can conceptually think that it's going to wear better, it's going to be more tear-resistant if it were a film. So you expect it to be longer lived. But it's not just what you expect, it's what you test and what you demonstrate to the FDA, because they're very concerned about actually proving safety and efficacy. Because if we go back to their mandate, which is actually in statute, it's to prove that the device is safe and as efficacious as the predicate device. And so for our example submission, our new device is a highly cross-linked ultra-high molecular weight polyethylene with a metal ceramic hip replacement device. The predicate is a linear. I've gone ahead and highlighted this. In this particular case, we want to use a reference device. And so a reference device is different from a predicate device in the fact that it's not demonstrating the same, it's not the same application, but it's using the same or similar technology. And so in this case, our predicate device is a hip replacement device. Our reference device is a knee replacement device. We're using the same material in the reference device as we are in the new device, but the predicate device is actually what the FDA is going to look at. They're also going to consider the reference device, but be very clear that the predicate device and the new device are the comparison across the device type. The reference device compares the material. And so I guess starting at the decision points that the FDA uses for this, is the predicate device legally marketed? It's going back to basics. Is the device that you're trying to compare this to, is it something that's on the market and ready to go? And so they prefer this sort of format where you take your new device, your proposed device, and compare that to the predicate device. And so legally marketed, to be determined. Hopefully yes. Actually, probably yes. Predicate device lists the K number for that. Go through the different classification names, provide the appropriate regulation, the product code, the device class, and these should all be a one-to-one match, with the exception of whether or not it's legally marketed right now, because we're going for substantial equivalence. Do the devices have the same intended use? They should in this case, because they are doing the same thing. The total hip replacement for both intended use and also the type of use, it's going to be prescription only. You're not going to be going down to your local pharmacy and picking this up for yourself. It's going to go into your hip and your doctor's going to put it there. It's prescription only, and it covers the appropriate CFR and regulation for that. And then specific to femoral heads and to hip implants, there are certain other aspects, for example, whether or not this is a cementless use only or whether it's a cemented. That's dependent on the guidance documentation for that particular device class. And then it also provides the indications for use, primary, secondary replacement, osteoarthritis, necrosis, basically anytime you have to replace your femoral head. Regards to decision point 3 and 4, do the devices have the same technological characteristics? And then secondly, do the different technological characteristics of the device raise different questions of safety and effectiveness? And so this is where we can start branching out a little bit. And so the different characteristics for this are effectively the same. We have wrought titanium, we have calcium phosphate stem coating, we have a cobalt chromoly femoral head coating on this. now at the acetabular liner, now we have a highly cross-linked ultra-high molecular weight, or an XLPE as the FDA refers to it, versus a linear ultra-high molecular weight. That is the only difference with regards to this particular medical device. And so now what we have to do is we have to answer no to do the devices have the same technological characteristics, but we can correctly answer the question, do the different technological characteristics of the device raise questions of safety and effectiveness? And the answer to that should be no. But then again, we'll also have to prove it. And so what we'll do is we'll do testing, which is prescribed either on the specific device's website or on through the judgment of the person submitting the 510(k) application to demonstrate that the data that we're collecting for demonstrating decision point 4 is appropriate. Are the methods acceptable? Does the data demonstrate substantial equivalence? So here are a few examples of physical properties testing that we would perform on the ultra-high molecular weight polyethylene and compare that directly to the linear polyethylene. For example, we might compare the tensile properties using ASTM E647. In this particular case, we would find that it conforms to the tensile properties that we expect and actually improves on the predicate. We might check fatigue crack growth rate through ASTM F2565, and we would see that that also conforms and improves on the predicate. Or we might compare the material properties using F2695. And again, it would be conforms, improves on predicate. And so the point is that the FDA is looking for information showing that the new device using the new material is as safe and as effective as the previous device, and so that they have no new concerns over safety or efficacy. And so with regards to decision points 5A and 5B, are the methods acceptable? Do the data demonstrate substantial equivalence? So far, we're thinking yes and yes. The next steps to this would be a head-to-head chemical characterization. And then following that, a risk assessment to identify any new or increased biocompatibility issues. And then a follow-up to that would be, do the material changes affect the device's performance specifications? And so a chemical characterization of this is what chemical species are present and how much of it are present. And so the first thing that we would do would be a literature review for observed chemicals for the toxicological concerns. And then an alternative would be, another point would be that the predicate and reference device must have the same processing and sterilization conditions. That's very important, because if you change the sterilization conditions, very frequently you introduce new vectors for concern. And there's an excellent webinar by the FDA where they go over this in great detail about sterilization. But the point of that is that your predicate and your reference device have to have the same things done to them to ensure that there are no new questions of safety and efficacy. So at Jordi Labs, how we have done head-to-head chemical characterizations in the past includes a host of techniques. And depending on your medical device, some of these are more appropriate than others. In this particular case, we would consider things like pyrolysis mass spectrometry, which provides qualitative polymer and additive chemistry for your sample. We might consider something like GC-LC-MS mass spectrometry, which would provide a quantitative and qualitative polymer additive chemistry. If we were concerned about the molecular weight distribution of your sample, we might consider something like gel permeation chromatography. ICP-MS is another technique which provides quantitative analysis of the inorganic components that are present, great elemental analysis with ICP-MS. And then alternatively, we might consider something like the functionalization or cross-link ratio using NMR, or potentially the filler content, although you can't necessarily use a ton of filler in these sort of applications. And so here's an example analysis where we performed PYMS, that's pyrolysis mass spectrometry, on an example analysis of polyethylene. And so it's a specialized technique that's intended for polymer analysis. It identifies not only the polymer structure and the additive package, but it also uses integrated Jordy proprietary and commercial libraries to do this sort of identification on the fly, and we're able to identify things that are very difficult to identify otherwise. And so you can see in the lower right-hand corner the pyrolysis mass spec of a polyethylene sample, and all the different oligomers that you're seeing are just fragments of that. And so we've compared, in this case, the predicate device and the de novo device, and they're very, very similar with the exception of some cross-linking. If we're interested in the nonpolar small molecules that are present in the sample, we might use something like QTOF GCMS. And this is an excellent case where we did several different extractions and we're able to identify, using the ultra-high mass accuracy of a QTOF instrument, to identify components that are present in the extract. And then on top of that, because we have that ultra-high mass accuracy, we can use very powerful algorithms to determine the molecular formula with scores and matches. And then add to that, we have, again, commercial databases as well as our own proprietary databases that we've accumulated over the years for reference matching different components. And so if all of that doesn't work, suppose you have some brand new molecule, we have structural elucidation through the fragmentation spectra. And so the odds are that we're going to be able to identify your unknown. And that doesn't just apply to GCMS results, that also applies to QTOF LCMS results for polar small molecules. We have the same mass accuracy with our QTOF instrumentation. We have the same structural elucidation with our QTOF. And so here's a few examples where we did positive mode and negative mode of a polyethylene sample for submission, and it turned out very nicely. Very few components to identify as expected for a medical device. With regards to analyte quantitation, this is where instruments like our triple quadruple mass spectrometry comes in handy. We have not only our triple quadruple mass specs, but also our CAD and UV vis signal for samples that don't have a ton of ionization. And so we're able to, in MRM mode, perform small molecule analyte quantitation, but also perform relative quantitation using, for example, UV vis or CAD. And so here's a trace of a CAD signal and a UV vis signal that came out of the same instrument at the same time, and we're able to quantitate each of the components that you're seeing there. Those different quantitations were conducted in this particular case using a relative quantitation strategy. As you can see here, this is a little bit of a complicated slide, but what you see along the x-axis are several different analytes. For example, ethylbenzene, styrene, it looks like stearic acid is present in there. And what you see in each case is 4 different bars, and those bars represent quantitation performed with different internal standards, whether it's deuterated toluene, deuterated naphthalene, etc. And so the point that I'm trying to make for this is selection of an appropriate standard is very important for this. It's very important to match the chemistry of your standard to the chemistry of the analyte that you're trying to quantify. Because in some cases you're going to vastly overestimate the presence of a component in the sample. In other cases, you're going to vastly underestimate. And so for components that are of medium toxicological concern or less, that is not especially toxic, it's often appropriate to perform relative quantitation because you can apply an appropriate safety factor in your toxicological assessment. On the other hand, for things that are very important to figure out exactly how much is present, we almost always recommend a formal quantitation. And so most 510K submissions are going to use a mixture of both relative and formal quantitation for the components that are not as, I don't want to say not as important, but not as toxicologically relevant versus the components that are especially of importance. And so the different strategies that we use for this sort of quantitation work, it may use an external standard, it may use standard addition. There are some drawbacks to formal quantitation where you absolutely have to have a commercially available high purity analytical standard to construct your calibration curve for this. And so there are trade-offs for each one of these sort of quantitation strategies. In all cases, we prefer to do formal quantitation. However, in some cases, it's not necessarily appropriate, depending on the analytical strategy you're looking for. Another way that we would be able to compare the different medical devices would be to do molecular weight distribution determination using gel permeation chromatography. And so what you're looking at here is several GPC traces of the linear polyethylene sample versus Unfortunately, the highly cross-linked ultra-high molecular weight polyethylene sample did not dissolve, and that's to be expected because it's a highly cross-linked system, but that's part of the submission that was going into the FDA. Another thing that the FDA is concerned about is, for example, the different trace elements that are present in the sample. And so here we have some ICP-MS results where we did quantitative elemental analysis for this. We have some pretty specialized instruments for this and analytical techniques for getting polymer systems to behave nicely with their ICP-MS. And for example, we have developed specialized techniques for dissolving polymer samples, which are notoriously difficult to get into an ICP-MS, and we also have an instrument that's specialized for high solids. And so in the same sort of analytical run, you can get trace elemental analysis as well as percent level elements to show up in the same analysis. And so, for example, in this one here, we detected no molybdenum, which is great. We detected no chromium, which is great, down to the parts per billion level. But we also detected a ton of zinc, which is acceptable because it's comparatively non-hazardous. Filler content's very important for a sample like this. In this sort of instance, we take a sample, heat it from ambient to 1,000 Celsius, and then we measure the weight loss as a function of time, and rather the temperature as well. And so they're very consistent, both the linear polyethylene sample as well as the ultra-high molecular weight cross-linked sample. They both had very low inorganic content, which was important for the FDA to find that. So the next steps for this, the FDA has a pre-submission feedback process where the FDA reviews your submission before you technically submit. There's no associated cost with the FDA. It does add between 75 and 90 days, but in most cases, our clients have been very happy with going through the pre-submission feedback process because it helps identify where the FDA may have concerns, and then we can get targeted and comprehensive feedback from the FDA before we file and have any sort of filing deadlines imposed on us. It gives you time to address any resulting concerns, address any additional performance testing, hopefully none needed, and then also provide appropriate justifications. And with that, you can file with confidence. And so here's a summary of the different techniques offered by Jordi Labs. It's pretty extensive. I won't go into detail on any of those, but just to let you know that we do a ton of different techniques. We have people who are experts in every one of these. And so our promise to you is that we'll provide modern chemical analysis, both with techniques and instruments, and we work with you to solve your problems. It's not data for data's sake, it's to solve your problem and provide the solutions you need. We make it our goal not to give up until we solve your problem, give the best data set possible for your project every time. And so with that, I wanted to thank you very much, and I'll take questions.